Hey guys, in this video, we'll be talking about regulation of energy balance. We'll talk about signaling, mainly with insulin and glucagon, and how that affects other pathways that we already learned in the cell. So this is a G-coupled protein receptor over here, the one on the left. This is a receptor tyrosine kinase. Those are the two receptor systems we'll talk about, mainly because glucagon uses G protein coupled receptors. Insulin uses receptor tyrosine kinase. So we'll talk about the mechanisms of these, and then we'll talk about specific ones for glucagon and insulin. And then we'll talk about how that regulates the different pathways in the cell. And then I also have stuff on microbiomes in our body at the end, because there's some questions on that. Okay. So organ communication. The whole point of this regulation of energy balance slides is it talks about how the whole body regulates itself. So it requires a lot of interactions between cells, organs, and systems, and we'll talk about that. And it's done through communication between organ systems. So this is done through the endocrine system. I'll go back to the other slide. The endocrine system is a collection of ductless glands that produce hormones to regulate metabolism, sleep, growth hormones. I'm sure you've learned about this already. Hypothalamus, pineal gland. This is where melatonin is made. Pituitary gland. I'm sure you've heard of anterior and posterior pituitary gland, thyroid gland, parathyroid glands, adrenal glands. Islets of the Langerhans in the pancreas. This is where you get your insulin and glucagon. You'll have your alpha cells and B cells, and then, of course, ovaries and the testes. So I'm sure you've somewhat seen this before. We're mainly going to focus on the pancreas in the slide because we're talking about insulin and glucagon. What these glands do is they produce these hormones that are able to travel. I'm sure you guys already know this, but I'll just briefly go over it. They produce hormones that are able to travel in the blood and then change transcription regulatory things in other parts of the body. So hormones, chemically diverse signaling molecules secreted by glands and transported in the circulatory system. They bind to the receptors on target cells, and these receptors are specific for whatever tissue we're in. So they're specific for the particular gland. Okay, so I'm sure you've seen all of this before. Just a review. This is the autonomic nervous system, which is also important part in the signaling. You have your parasympathetic and sympathetic. Parasympathetic is your rest and digest, so that's after you eat a meal. Your body will constrict its pupils, slow heart rate, constrict airways, and then stimulate activity in the stomach. And then fight or flight, this is when you're and parasympathetic releases acetylcholine. I don't think you need to know this, which acts on other cells. Sympathetic releases norepinephrine and epinephrine. Those are released from the adrenal medulla, and then they mediate the sympathetic activity. You die. So this happens if you're like someone's attacking you, you're in danger. Sympathetic fight or flight. You dilate pupils, inhibit salivation, increase heartbeat, relax airways, inhibit stomach activity, and basically the opposite of what parasympathetic is. This is important in our signaling, which is why I mentioned it. Okay, so hormones, we already mentioned this, they circulate in the blood and bind to specific receptors. So you have your endocrine cells here. This is the bloodstream. They release the hormones in the blood, and then the hormones are able to bind to whatever cells express the receptors that they're looking for, which is tissue specific. Okay, so all this stuff should be reviewed from bio, but I just included it because it introduces the topics we're talking about. Okay, so the two main kinds of receptors we have, we'll go over both. We have the receptor tyrosine kinase, and then we have G protein coupled receptors. Okay, so for receptor tyrosine kinase, this is what insulin will use. So when we talk about insulin, we'll go over the mechanism of the receptor tyrosine kinase. We also have the G protein coupled receptor. and this is what glucagon uses. So an easy way to remember this, usually I remember G protein coupled receptor starts with a G and glucagon starts with a G. So then I know glucagon uses G protein coupled receptors. So that means insulin has to be the one using receptor tyrosine kinase. So that's kind of an easy way that helped me remember it when I was learning this. Okay. So why do we go through this? Why do we bind receptors and why do we release hormones to have effects on other parts of the body? A big reason is due to amplification. So in your cell, you have a limited amount of receptors, and you want to get the most out of those receptors to change the signal inside the cell. So in these receptor systems, you have what is called the signaling cascade. So something activates something, which activates something else, which activates something else. So this allows us to amplify the signal. So for example, when we talk about adenylate cyclase, which makes cyclic AMP, so when in G protein coupled receptors, when one adenylate cyclase is activated, we're able to make multiple cyclic AMPs. So for one adenylate cyclase, we got we got like 100 cyclic AMPs. Okay, so we're able to amplify the signal. So now these 100 cyclic AMPs can activate, they can go on and activate PKA, for example, so they can activate two PKAs or something like that. So you basically just amplify the signal as you go on. So activation of V receptors on the outside of the cell is amplified in the internal cascade. This is important because receptors on the outside cells are usually in low abundance, and you want to get the most out of those receptors. So that's why this signaling cascade mechanism was evolved. Okay. So signaling, which we'll talk about. So when we activate the receptor on the outside of the cell, it will activate other things in the cell. It uses a lot of mechanisms when it activates stuff in the cells. A big one, which we've already seen with post-translational modification, is going to be phosphorylation or dephosphorylation. So when you activate something, you're usually going to phosphorylate it inside the cascade. Usually, most of the time. If you have no idea, you can say that's a good guess, but sometimes it's the other way around. And then usually when you inactivate something, you dephosphorylate it. The three amino acids that are usually phosphorylated are your serine. These are your ones that have the OH on them. Serine, threonine, and then tyrosine. So usually 99% of the time, those are going to be the one phosphorylated. This is important for the MCAT as well if you're studying for that, to know what three amino acids get phosphorylated. So you can have phosphorylation, dephosphorylation. You can also have protein interactions. This usually occurs with dimerization. So you have like protein A, protein B, and when they interact, they dimerize. This can activate another part of the signaling cascade. Okay. So this is the receptor tyrosine kinase overview. Sorry about that, I was just plugging in my charger. Okay, this is the receptor tyrosine kinase overview. So insulin uses this, but insulin is not the only thing in your whole body uses this. A lot of molecules can use the receptor tyrosine kinase, but insulin is usually the example that is taught when we learn about the receptor tyrosine kinase. So insulin is your ligand. Once it finds the receptors, it will bind into these two alpha subunits. This induces dimerization in this subunit and this subunit. So each subunit has an alpha and a beta. Once it dimerizes, it autophosphorylates itself on cytoplasmic domains. So if we look at it, this is the extracellular space. So this is what binds the insulin. You have your cell membrane, and you have your cytoplasm. When the insulin binds on the extracellular space, this induces dimerization and then eventually autophosphorylation in the cytoplasmic domain. This autophosphorylation occurs on tyrosine residues, and it's done by a kinase that the enzyme has. Okay, so this is pretty much everything we'll go over, but we're going to break it down briefly, but this picture has everything. So we have receptor tyrosine kinase, insulin would be over here. We would bind, this would cause dimerization, and then tyrosine residues would get phosphorylated. You have stuff called SH2 proteins. They recognize the phosphorylated tyrosine residues, so they bind to them. They bring along with them another type of proteins called SH3 and a protein called SOS. SOS is usually bound to an inactive RAS, which has a GDP bound to it. RAS can be activated by an enzyme called GAP. Once RAS is active, it has GTP. It phosphorylates RAF, and it can also phosphorylate other types. So this is one type of mechanism. Remember, there's different types of these. Once RAS is activated, it can activate RAF. RAF can activate MEK. MEK can activate MAPK. And then MAPK can enter the cell and phosphorylate other stuff. So this is one way that receptor tyrosine can go. There you can have different levels, especially down here at these kinases. So briefly just go over this. Phosphorylated residue can assemble signaling cascade protein via SH2. So this is talking about the SH2. This recruits other cytoplasmic domains. This is SH3 and SOS. And then you eventually activate small G protein coupled receptors, which we'll talk about in a sec. So that's your RAS. RAS is a group of small G protein coupled receptors. You have a lot of them. And then this initiates a cascade of phosphorylation events and kinase activation. So this is talking about the RAS, RAF, and MAP. And then the phosphorylation eventually the transcription factors. Transcription factors can enter the nucleus and then change gene expression of the cell. Okay, so let's talk about SH2 and SH3, which are right here. Sorry, I kind of just went over like everything without breaking it down. So SH2 and SH3, those are considered small protein molecules. This is the DNA segment of them. They're transcribed together. SH2 is the one that recognizes the phosphotyrosine. And then SH3 helps link the surface proteins. So this is talking about like the surface proteins is considered the receptor. And then with the intracellular signaling. So that would be binding to SOS, allowing SOS to bind RAF. Or not RAF, RAS. Yeah, it binds RAS first. And then it's made of proline and hydrophobic amino acids. So this is their DNA. They have the catalytic domain, and then they're usually transcribed together. And then this shows that when SH2 recognizes the phosphorylated tyrosine, SH3 come along with it, and then they'll able to initiate other things in the cell. Okay, so small G proteins. This is a large superfamily, but the ones we'll talk about are going to be RAS. So when RAS is inactive, it has GDP bound to it. When RAS is active, it has GTP bound to it. Okay, I think this picture has P21 instead of RAS. So this is would be another example of the small G proteins, but usually you're tested on RAS. So you can just think of the same thing, but in terms of RAS. So inactive RAS has bound GDP bound to it, and active RAS has GTP bound to it. Guanine exchange factor GEF activates RAS, so it takes this. We hydrolyze a GTP. The phosphate goes on RAS, and then you get your GTP, and then GAP stimulates GTP hydrolysis. When it does this, it inactivates RAS, and then you're able to activate when you hydrolyze the GDP. Your next signaling protein. So an example would be RAF. So when RAS hydrolyzes GDP, you inactivate it, and then you activate the next signaling molecule, which is RAF. And then active RAS binds effectors and activates them. Okay. So, uh, like I said, RAS, RAF, MEK, MAPK, all those stuff, they're just one example of different things in the cell. Okay. So I don't think you're expected to know this, but this is allowing you to see the different signaling cascades that can occur. So for growth factors, RAS eventually activates RAF, which is a type of M kinase. And you can also activate other types of RAF, and then you have MOS. Then those can activate a type of M kinase, which MEC1 and MEC2 are example of, and then M kinase can activate MAP kinase. An example of MAP kinase would be ERK1 and ERK2, and then ERK1 and ERK2 can activate transcription factors which eventually induce growth differentiation. So this is your growth factor. You have one for stress. You have you pretty much have one for a lot of things in the cell. So I'm not sure, but I don't think you're expected to know all of these. I would know the RAS, RAF, MEP. Let's go back. Yeah, this one. RAS, RAF, MEC, and then MAPK. And then for MAPK, you can do ERK1 or ERK2. It doesn't matter. Because ERK1 and ERK2 are a type of MAPK. Okay. So yeah, multiple kinases activate each other. So you have these are kinases. They activate this, they put a phosphate on it. This activates the next one, puts a phosphate on it. And then you just go on. Then eventually you activate transcription factor, which is able to change transcription inside the cell. Okay. So now we talked about receptors, tyrosine kinase. Let's talk about GPCR. So it has 7 transmembrane domains. So transmembrane domain just means they're in the membrane. So you have your GPCR, you have your transmembrane protein, and then you have heterotrimeric G protein. So you have your G alpha, G beta, and G gamma. Okay. G alpha, that's mainly going to be your thing that's going to go on and activate other things. In the inactive state, it's bound to GDP. And then once a ligand binds and it gets activated, it dissociates, and then it has GTP bound to it. And then it can go and phosphorylate other things in the cell. G beta and G gamma can actually do some signaling of their own, but usually their job is to hold G alpha when it's bound to G DP. G alpha, we can have different G alphas. There's G alpha S, which means it's stimulatory. So this G alpha would stimulate adenylate cyclase, which is we'll talk about in a sec. G alpha and I, which is G alpha inhibitory, so it would inhibit the next signaling cascade. You can also have G alpha Q, which we'll talk about briefly. So what happens is when a ligand, so for example glucagon, so like I said, glucagon uses this, but glucagon is not the only thing in the world that uses this. Other molecules can use this system, but usually when we learn about it, we learn it with glucagon. So when glucagon binds, G alpha, instead of being in the inactive state, it dissociates and it grabs a phosphate, so it has a GTP, and then it's able to hydrolyze that GTP and activate the next molecule or the next protein. Okay, so what happens when glucagon binds? So this will briefly talk about what I just mentioned. The ligand binds GPCR. G alpha first exchanges GTP. This should say GDP to GTP. So it exchanges GDP to GTP. Here, I'll do this. GDP to GTP. G alpha dissociates from G beta and G gamma. Then G alpha hydrolyzes GDP and activates adenylate cyclase, which is the next protein in our signaling cascade. And then once it activates adenylate cyclase, it reassembles and then binds GDP. And then it waits until another ligand binds. And then G beta and G gamma can do some signaling of their own. So here we have the ligand. It would come in, bind a GPCR. This is G beta, G gamma. G alpha is bound to GDP. G alpha would dissociate. And then it has GTP bound to it. This is adenylate cyclase. And then it binds, activates adenylate cyclase, and then we'll talk about adenylate cyclase in a bit, but adenylate cyclase can make cyclic AMP and IP3. Okay, this is the structure of G alpha. Okay, adenylate cyclase. So this is the protein that is activated by G alpha. So it's a membrane-bound enzyme, so it's in the membrane. Takes ATP and converts it into cyclic AMP. Okay, so this is cyclic AMP over here, and this is ATP. When we convert that, we lose a pyrophosphate. Okay, so cyclic AMP is different from AMP. Okay, this is a common misconception. If you have high AMP, you would think you have high cyclic AMP, or if the other way around, it doesn't work like that. So, the thing we will talk about in this lecture a lot is what happens when receptor tyrosine kinase is activated or G protein coupled receptors. So insulin, right? Glucagon. Insulin is released after you eat, so you have high blood glucose levels, right? So insulin is released after eating a meal, so you have high energy. Glucagon is released during low blood glucose levels. Okay, so low energy. It's important to look at these and help us answer questions and make sure it makes sense at the end, because you just don't want to memorize a bunch of pathways if they don't make sense. So insulin is high blood glucose levels. So we want this eventually to activate glycogen synthesis, right? Because we have a lot of glucose. We'll probably want to run glycolysis, maybe some fatty acid synthesis. We can do cholesterol synthesis if we want. So basically stuff that is run when we have high energy. This is run when we have low energy. So gluconeogenesis, because we have the liver would run gluconeogenesis because we have low blood glucose levels, so it needs to make glucose and send it to other tissues. We would probably run beta oxidation to break down fats for energy. We probably break down glycogen. Breakdown. You get the point. But so this happens when glucagon binds. Glucagon uses GPCR, and eventually we make cyclic AMP. Okay, so you see that the cyclic AMP pathway that activates is very similar to what AMP activates, right? Because AMP is the low energy, but they don't correlate together. We'll see why AMP eventually activates very similar things to cyclic AMP, but they act on different things in the cell and they're different from each other. But the pathways they act on eventually lead in the same direction. So cyclic AMP will eventually activate gluconeogenesis, beta oxidation, and glycogen breakdown. AMP will probably do the same thing, but they're not related to each other. Hope that makes sense. Hopefully it didn't confuse anyone. And we'll talk about what AMP acts on in a sec. And in the next slide, I'll talk about what cyclic AMP does. Okay, so adenylate cyclase, it takes ATP and it makes cyclic AMP. It has a magnesium ion, and then we lose a pyrophosphate. Okay. So once we make cyclic AMP can go and activate a protein called protein kinase A, which is serine and threonine kinase. That means it's a kinase that phosphorylates these two residues. So it has two regulatory domains and it has two catalytic domains. So we take 4 cyclic AMPs, they bind to the two regulatory domains. This allows the catalytic domains to become active and freed up, so then they can go and activate the next event or the next protein in the signaling cascade. So eventually this will cause a downstream phosphorylation of events. Okay. So we had glucagon bind, GPCR, G alpha dissociated, activates adenylate cyclase. Adenylate cyclase made cyclic AMP. Cyclic AMP activated pKa. Okay, so let's talk about what pKa can do. So kinases are usually multifunctional. They can have different effects. So one thing pKa can act on is cyclic AMP dependent protein kinase. This eventually will act on PFK2, which we'll talk about in later slide more specifically. pKa can phosphorylase kinase. Phosphorylate kinase phosphorylates glycogen phosphorylase. Okay, what does this mean? So if we remember when we talked about glycogen phosphorylase, right? Glycogen phosphorylase is what breaks down glycogen. Phosphorylase kinase puts phosphorylase in the active state. So it phosphorylates it, so it's able to break down glycogen. Okay, so this phosphorylase kinase. pKa activates the activator. Okay, so because it activates the activator, we get more glycogen breakdown. And this kind of makes sense, right? Because when we release glucagon, we're in a low energy state and we need to make break down things somehow. So this pathway makes sense. And I would recommend checking these whenever you're talking about glucagon and insulin to make sure that the pathway you're activating is being activated or inhibited in the right direction. So when we have low energy, we want to break down glycogen, yes. So this makes sense. Okay, pKa can activate ion channels as well, and it can also act on transcription factors. So you have the cyclic AMP response element. It can also activate transcription. We'll mainly talk about pKa and cyclic AMP. So this is the not pKa. We'll mainly talk about phosphorylase kinase and cyclic AMP dependent protein kinase. Okay. So that was G alpha S. So G alpha S stimulates adenylate cyclase. You also have G alpha. Remember I said there's different G alphas. G alpha S inhibits. G alpha I inhibits adenylate cyclase. Okay. So you have G alpha Q. So G alpha Q is different. It activates a different pathway. It doesn't even act on adenylate cyclase. It activates a protein called phospholipase C, or you can call it PLC. When PLC is activated, it cleaves a membrane protein called PIPE2. When it cleaves this, you get IP3 in a protein called DAG, or not protein, just a molecule. Yeah, it's a protein. And then both help to release calcium from the sarcoplasmic reticulum. When calcium is released, the calcium comes in and DAG, and they both activate a different protein kinase called protein kinase C. So this is different from protein kinase A. Protein kinase C can then go activate gluconeogenesis or glycogen breakdown. So remember, we have to check if this makes sense. Glucagon is what uses G protein coupled receptors and eventually activate G alpha Q. Do we want to activate gluconeogenesis and break down glycogen when glucagon binds? Yes. So that means we memorize this pathway correct. So it's important to check this when you're going through the different pathways. Okay. So G alpha Q activates PLC. PLC cleaves membrane PIPE2. You get IP3 and DAG. Which both help release calcium. Calcium and DAG come in, activate protein kinase C, and then protein kinase C can act on gluconeogenesis and glycogen breakdown. Okay, this is a different protein, completely different. It's called AMPK. We already talked about this, so this is a master regulator. It's called AMPK kinase, pretty much. It's activated by AMP, which is comes in low energy. And it's inhibited by ATP, and they're mutually exclusive, so you can't have both binding. It's either it's inhibited or it's activated. We have a G alpha beta heterotrimer. So hetero just means they're different, trimer just means 3 subunits. The alpha subunit is the catalytic subunit, beta subunit is the regulatory subunit. Okay, so AMPK, we already talked about it. Basically, it's activated by low energy, so we'll activate things in the cell that eventually give us more energy. Okay, so when AMP binds, so this is low energy, when it binds, it will probably activate AMPK. It changes the gamma subunit, and it allows it to increase phosphorylation. So AMP binds, and then the gamma subunit has a conformational change, and it allows it to be phosphorylated more commonly. This phosphorylation increases the activity of the protein 100 times. Phosphorylation occurs at residue threonine 172. By the way, this information, like, you'll never be tested on it, what residue it's phosphorylated on, but if you have an extended response, a lot of the times the extended response is going to be greater than how much you know. If you were asked this question and you're able to tell them, oh, it was phosphorylated at threonine 172, it will show more understanding. So the grader will be more lenient when they're grading these, because you show that you studied, you know your material very did the exact point. Okay, so 3 AMP binds, changes the conformation, and then we're allowed to be phosphorylated at threonine 172. This can be done by 3 kinases. So you have LKB1, P1, CAMKK2, and then TAC1. Okay, so all of these would come in and activate AMPK once AMP binds. PKA, so let's talk about PKA. Where does PKA come from? So this comes from glucagon. Do we want to activate something that eventually leads the AMPK being activated in terms of glucagon? Okay, well, glucagon is released when we have low energy state, when we want to break down glycogen, when we want to break down fatty acids or do gluconeogenesis in the liver. And AMPK is very similar, right? AMPK is activated in low energy. So PKA, so this is the glucagon pathway, can activate LKB1. So it can help activate AMPK. So I know I said cyclic AMP and AMP, their levels are not related, but because cyclic AMP activates PKA and then PKA can activate AMPK, without AMP, this won't happen. And cyclic AMP levels don't depend on AMP. So they're kind of related in somehow, but the the concentration of cyclic AMP and AMP are not related at all. So if you have high cyclic AMP, that doesn't mean you have high AMP or vice versa. Okay, so AKT, which is PKB, so we talked about PKA and PKC. PKA and PKC are in the GPCR. PKB is in the insulin pathway, which we'll talk about in a second. So insulin does opposite of glucagon. So insulin probably wants to inhibit these 3 kinases. Another name for PKB is AKT. Okay. So what does AMPK do? So basically it's activated by AMP, so stuff in low energy, and it's inhibited by ATP, high energy. So this is able to bind AMPK. AMPK then activates ATP producing pathways, so AMPK will probably activate glycolysis, beta oxidation, glucose uptake. I don't think they have it here, but glycogen breakdown as well. We just want energy somehow. We'll also inhibit ATP consuming pathways, so we'll inhibit fatty acid synthesis, cholesterol synthesis, glycogen synthesis, and protein synthesis. All the stuff that requires ATP. Okay? A little different than from what glucagon does, because glucagon activates gluconeogenesis, AMPK activates glycolysis. So they're slightly different, but it's important to be able to relate them to each other. Okay, AMPK on ACC, so we already talked about this. ACC, so this is first enzyme in fatty acid synthesis. And then ACC2, so this is the one in the cytosol. ACC2 is the one in the mitochondria, and it's the one that makes malonyl CoA to inhibit CPT1. Okay, so do we want to run fatty acid synthesis when AMPK is activated, when we have low energy? Probably not. So we will inhibit fatty acid synthesis to inhibit ACC1 and ACC2, and then we'll upregulate beta oxidation when we do that. Phosphorylated ACC is inhibited, so AMPK comes in, adds a phosphate on ACC, and then it becomes inactive. So remember, because we're inhibiting ACC2, this allows CPT to run forward without being inhibited, so this will upregulate beta oxidation in the process. Okay, AMPK on glycolysis and gluconeogenesis. So remember, AMPK is activated by AMP, so low energy. So we probably want to activate glycolysis and inhibit gluconeogenesis. So AMP activates AMPK. AMPK then phosphorylates PFK2 and activates it. So if you remember from glycolysis at all, PFK2 makes fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate can activate PFK1, so this is rate limiting step in glycolysis. It also inhibits fructose 1,6-bisphosphate, so this is the rate, one of the rate limiting steps in gluconeogenesis. Okay, so it will activate glycolysis and inhibit gluconeogenesis. Fructose 6-phosphate activates PFK1 in glycolysis, inhibits fructose 1,6-bisphosphate in gluconeogenesis. This is also showing you AMP directly acts on PFK and ATP as well. AMP activates PFK and ATP activates fructose 1,6-bisphosphate. Okay, so this is all from glycolysis. Okay, so we talked about acts on fatty acid synthesis and glycolysis. So let's talk about hormonal control. So remember, we said insulin is released at high glucose levels and it tries to lower it. So the pathways insulin binds tries to lower blood glucose levels. Glucagon is released at low blood glucose levels and it tries to raise it. So what does insulin tell the cell? It tells the cell that you have glucose and amino acids available. Prepare for substrates. So insulin just tells the cell that, hey, we have a lot of substrates that we can use to make energy. Glucagon does the opposite. It tells you have glucose and amino acids are low. We need to get substrates from the cell and put them in the blood and give them to the organs that need them the most. So glucagon does the opposite. Epinephrine is very similar to glucagon, except this is usually is released by adrenal medulla and activated by sympathetic nervous system. So this would be like fight or flight when you would release epinephrine. So in fight or flight, you have the same needs as glucagon, but you need the substrates immediately. It needs to happen quicker. So that's what the difference between epinephrine and glucagon. Glucocorticoids and thyroid hormones, they tell the cell glucocorticoids changes these substrates long term, so that's glucocorticoids. Then thyroid hormone increases basal metabolic rate. Both of these, because they act long term, they change the gene transcription of the cell. Okay, so because they act long term, they need to change the gene transcription of the cell. Okay, insulin and glucagon. Insulin is made in beta cells of the pancreas. Glucagon is made in alpha cells of the pancreas. This is a picture of it. So you see alpha cells make glucagon, beta cells make insulin. You also have delta cells, which make somatostatin. Somatostatin helps regulate between insulin and glucagon. You don't have to know about somatostatin, but it's in the picture, so I'll talk about it. Insulin is 110 amino acids, and glucagon is 29 amino acids. Okay, let's talk about how high blood glucose levels eventually lead to insulin being made in beta cells. Okay, So start over here. So this is glucose. You have high blood glucose levels. Enters the beta cells. So this is beta cells. Goes through glucokinase. Yeah, glucokinase. Because the beta cells also use glucokinase, not hexokinase. You make glucose 6-phosphate. And then you pretty much go through glycolysis. And then eventually make pyruvate, make the Krebs cycle. Get your electron carriers. They go to ETC. You make ATP. So ATP rises. When ATP rises, this triggers closing of calcium channels, and in the process, Na and calcium channels open. Okay, so calcium channels, they have calcium has high concentration in inside the cell and low in outside. So if calcium channels are open, it allows positive calcium channel to leave the cell. Okay, so if we inhibit this, right, we're inhibiting positive molecules leaving the cell. So this will allow inside the cell to be more positive. We're also, so sodium and potassium, sodium and calcium are different. They're high outside and they're low inside. So they want to enter. And you have L-type calcium channel, D-type, T-type. You don't have to know the difference. So we close potassium and we open N, sodium and calcium. Those ones allow to come inside the cell. This results in more positive cells inside the cell because we retain the calcium and then we allow sodium and calcium to enter. We retain the potassium, we allow sodium and calcium to enter. And then this causes depolarization. Okay? Eventually, the calcium that's also entered the cell triggers release of vesicles containing insulin. So you get your action potential, this extra calcium comes in, and then you have a reserved pool of insulin stored. Calcium acts on it, and then insulin is released from outside the cell, and and then it's able to go and bind on organs to tell them that, hey, we're in a high energy state. We have a lot of glucose. So this is what happens inside of a beta cell. Okay. So this is the insulin receptor. We already talked about the receptor tyrosine kinase. So it's a dimer. It has IRS1 and IRS2. So IRS1 is here, and IRS2 is here. When insulin binds, dimerizes, autophosphorylate cytoplasmic domain residues of tyrosine, and then other proteins are able to bind it up. Okay. IRS1, IRS2, they're scaffold molecules. They have what is called the PTB domain. This is where the tyrosine gets phosphorylated. Okay, so this is the gene of IRS1, and this is the different domains that it has. This gene, PTB, when it's translated, well, you transcribe it first, mRNA, and then you translate it. Eventually, when it's translated, you'll have a bunch of tyrosine residues that are able to get phosphorylated. Okay. Once they're phosphorylated, this recruits SH2, and then we haven't talked about this yet. Protein phosphoinositol, I think that's how you say it, 3 kinase or PI3K. So you recruit these two molecules to bind to the phosphorylated tyrosine. Okay. PI3K phosphorylate phosphatidylinositol lipids in the cell. And then, so this is the pathway over here. You have insulin, IRS, PI3K, SH2 is also over here. We activate a bunch of things. I don't think you guys are expected to know that, but eventually AKT or PKB gets activated, and then this can act on cellular targets. Additional pathway is using SH2 and SH3, and then this is the one where we talked about at the beginning, activates RAS, RAF, MAPK, and then changes gene transcription. So insulin can use this pathway, activate PKB, and PKB can act on cellular targets, or it can use the RAS, RAF, MAPK pathway and then activate gene transcription. So insulin can do either way. Like just like we talked about glucagon binding through G protein, different types of G protein coupled receptors, G alpha Q, G alpha S. Insulin has similar mechanisms. Okay, so PKB. So remember, when is this activated? So this is activated when insulin is released. So we have high blood glucose level. So we keep that in mind. So activation of PKB, AKT. So this inactivates glycogen synthase kinase 3, which keeps glycogen synthase active. So in summary, it stimulates glycogen synthesis. Okay, so glycogen synthase kinase, right? It adds phosphates on glycogen synthase and it inactivates it. So this inactivates it. Okay, what AKT or PKB does, so let's do PKB, is it inhibits the inhibitor. So we eventually put glycogen synthase in the active form. And this kind of makes sense. If we have a lot of PKB, that means insulin is binding. And then when insulin is binding, you want glycogen synthase to be active. So this is another way to look at it. This is receptor tyrosine kinase. You have IRS1. It becomes phosphorylated tyrosine. PI3K, SH2 come in and bind to the phosphate. This eventually activates pipe 2, and then you get pipe 3. Pipe 3 then activates, we didn't talk about this part, activates PKB. Then PKB puts glycogen synthase in the inactive state. And then when glycogen synthase is in the active state, glycogen synthase kinase, sorry, glycogen synthase kinase is in the inactive state. This keeps glycogen synthase in the active state. Okay. Okay, so now we talked about how glucose stimulates insulin release in beta cells. Let's talk about how insulin mediates glucose uptake in other cells. So insulin signals to the cell that we have a lot of glucose, and the way that it does this is it allows the cell to insert GLUT transporters into the membrane. So the one we're talking about is GLUT4, but it does this with all the different GLUTs as well. This allows glucose to enter the cell. GLUT4 transporters are usually inside vesicle walls. So remember we said insulin activates PKB and AKT. PKB goes in, phosphorylates cytoskeleton proteins to promote translocation of vesicles containing the GLUT4. So the vesicles are containing GLUT4. These vesicles go in the membrane and then insert the GLUT4 transporters. When you have more transporters, you'll have more glucose entering the cell, so you increase glucose uptake. Okay, so let's talk about insulin's effect on glycolysis. So remember, insulin is high blood glucose levels, so we probably we probably want to stimulate glycolysis because we have a lot of glucose stored in the cell, or stored in the blood. So we want to use that glucose somehow, so the cell can use glycolysis to get rid of it and make energy for the cell. PKB, which is activated by insulin, we already mentioned that. It activates phosphoprotein phosphatase, which we'll talk about briefly. Phosphoprotein phosphatase activates PFK2. PFK2, remember, makes fructose 2,6-bisphosphate, which stimulates glycolysis. Okay, so if we go look here, we see that phosphoprotein phosphatase, which is the one activated by AKT or another name for PKB. Phosphoprotein phosphatase takes a phosphate off PFK2, puts it in the active state, and then PFK2 goes in, makes fructose 2,6-bisphosphate, which acts on PFK1. When PFK1 is upregulated, glycolysis is also upregulated. Okay, so that's what insulin does. So insulin stimulates glycolysis. Okay, cyclic AMP. So remember, cyclic AMP is released from adenylate cyclase. Adenylate cyclase comes from the glucagon pathway. Glucagon eventually makes cyclic AMP. Cyclic AMP activates cyclic AMP dependent protein kinase. So we briefly talked about this in the beginning and how PKA can act on it. So cyclic AMP can bind that and activate it. and it inhibits PFK2, so no fructose 2,6 bisphosphate is being made, which stimulates gluconeogenesis. So if we look here, phosphoprotein phosphatase puts PFK2 in the active state, and then cyclic AMP dependent protein kinase puts PFK2 in the inactive state. So it does the opposite of the other one. And it kind of makes sense because you want glucagon and insulin to do the opposite things. This is different from AMPK and how AMPK acts on PFK2. So we already talked about how PFK2 activates. AMPK activates PFK2. So this is different from it. So remember, AMPK phosphorylates PFK2 and makes it active. Okay. We just said that phosphoprotein phosphatase dephosphorylates PFK2 and makes it active. So what I want you to take away from this is a phosphate group on a protein doesn't mean it's activated or inactivated. You have to look at what is going on with it. So PFK2 can be active in the phosphorylated or the dephosphorylated state. And that's why it's important to distinguish between AMPK and the pathways in insulin and glucagon. Okay? And yeah, that's why I put this happens in the liver, because it also depends on the tissue that you're in. This is talking about the liver. When we talk about AMPK, it's usually in other tissues as well. AKT, AMPK are different things that affect PFK. So yeah, I mentioned that AMPK and PKB, different things that eventually affect the mechanism of PFK. Okay, so glucagon and epinephrine. Epinephrine, norepinephrine, very similar. Both released by the adrenal medulla. We don't have to know much about epinephrine. Released by adrenal medulla, stimulated by sympathetic nervous system, it helps with the fight or flight. Glucagon is a peptide hormone. Epinephrine is a small molecule synthesized from tyrosine. They both use G protein coupled receptors. Glucagon uses glucagon receptors, and epinephrine uses I could never pronounce this word. Adrenergic receptors. They both bind and eventually stimulate adenylyl cyclase to make cyclic AMP, and then cyclic AMP goes and activates PFK. Or not PFK, PKA. Two different things. All right. So remember we said glucagon and then epinephrine. They both activate this enzyme, and then this is the different things that PKA can act on. So remember, this is telling the cell that we're in the low blood glucose levels, right? So what things do we want to run? We probably want to run like gluconeogenesis. We probably want to run. We probably don't want to run fatty acids synthesis. We want to run beta oxidation. We don't want to run glycolysis. So we just want to run stuff that makes energy for the cell or makes glucose to upregulate blood glucose levels. So glycogen synthase. Glycogen synthase uses glucose to make glycogen. So we probably want to reduce the activity of that since we have low blood glucose levels. So yeah, free glucose is not available to be stored. Phosphorylase kinase. So what is phosphorylase kinase? It activates the phosphorylase enzyme. So it puts a phosphate, phosphorylase kinase. Let's go back. Did I skip it? Yeah, no, phosphorylase kinase makes phosphorylase active, and then we're able to break down glycogen more efficiently. Okay. So we probably want to activate phosphorylase kinase to be able to break down more glycogen. PFK2. So PFK2 activates PFK1. Do we want to run glycolysis when we have glucagon binding? Probably not. So we'll probably reduce activity of PFK2. So PFK1 is less active. And we're able to inhibit glycolysis and increase gluconeogenesis because that's what we want when we have low blood glucose levels. Fructose 2,6 bisphosphatase, so this is opposite of PFK2. Will probably increase the activity of this to inhibit glycolysis and increase gluconeogenesis as well. Hormone-sensitive lipase, because we have low blood glucose levels, we'll probably look for another energy source that the cell can use. So we'll activate lipases so they mobilize fats and break them down. Okay. So this we'll talk about insulin in diabetes in a bit. But this is just a map from the CDC, shows that obesity and diabetes, so this is their occurrence in the United States, and they tend to be very correlated. So type 1 diabetes is autoimmune, so this is you destroy your beta cells, so you're not unable to make insulin secreted. So usually treatment for this would be give the patients insulin injections. Type 2 diabetes is your receptors don't respond to insulin. Okay, this usually happens at younger age. That type 1. Then type 2 is older, and that's the one that's usually correlated with obesity. Okay, what happens during diabetes? So remember diabetes, so we'll talk about this happens for type 1 and type 2. So you have your reduced insulin response. So type 1 is you don't have insulin at all. Type 2 is your insulin receptors don't work. So either or you'll have less insulin response. So you won't be able to activate PKB, and then PKB won't be able to act on different things that it does in the cell. When this happens is the cell will eventually release a lot of glucagon because insulin is not having its effect. So even though you have blood glucose levels, it will tell the cells that you have low blood glucose levels because the glucose is in the blood and it's unable to get inside the cell. So excess glucagon accumulates. Epinephrine also accumulates. They bind to the receptors, eventually activate adenylate cyclase. Adenylate cyclase takes ATP, makes cyclic AMP. Cyclic AMP is, remember, cyclic AMP released from glucagon, so it will result in less glycolysis and increased gluconeogenesis. Will activate lipases, so we break down fats, tag breakdown, and then in the blood. So we'll break down lipases we'll activate lipases to break down tags, so increase fatty acid concentrations in the blood. So yeah, the glucan activated pathways will be upregulated. Okay. Glucose in the blood. So in insulin, because insulin is not binding, even though you have high blood glucose levels, the glucose is unable to enter the cell. So glucose inside the cell will just continue to accumulate and not be taken up by the cell. This will result in decreased in glycolysis and glycogen synthesis because we have the glycogen is not, or not the glycogen, the glucose is not entering the cell. And this will result in proteins inside the cell being degraded to make glucose. It will also increase fatty acid oxidation. Okay, so insulin resistance. Let's talk about how that relates to the whole body in general. So in fats, when you have insulin resistance, the liver will signal like, hey, we don't have any glucose in our tissues, right? Because insulin is not binding, even though there's a lot of glucose in the blood level in the blood, but the body is unable to detect that. So it will signal the fat cells or fat tissue to increase lipolysis, so to break down fats. This increased fatty acids in levels in the blood can go to the skeletal muscles and they can go to the liver. They go to the liver. Let's follow the liver path. And they increase glucose production. This glucose production in the liver made, so the liver is like, hey, we have really low blood glucose levels because glucagon is the only thing that's binding. So the liver takes the fats and turns it into more glucose and puts it in the blood. So it results in more glucose in the blood, okay? The fats, they're like, hey, we have low blood glucose levels. Like, my job is to store fats. I probably shouldn't take up glucose from the circulatory system and leave the glucose in the blood for the organs that need it, even though there's a lot of glucose in the blood. So insulin is not able to bind to the fat tissue to mediate glucose uptake. So this results in decreased glucose uptake, which again contributes to hyperglycemia. Again, fatty acids also go into the skeletal muscles. Muscles is able to use that for energy, but because insulin is not able to bind on skeletal muscles, it's unable to mediate glucose uptake. So this results in decreased glucose uptake as well and more clearance because skeletal muscle will clear it to other tissues that need it. So will also release in hyperglycemia. Okay, so another thing that the liver will do is we will make VLDL molecules. So you remember VLDLs? They take they take fats from the liver and send it to other tissues in the body that need it. Okay, so the liver's like, hey, we're like insulin's not binding. We have a lot of glucagon. Other tissues are probably starving. They need that, so they increase VLDL production to send uh, fats to other parts of the body. So this actually, even though they don't need the fats, this results in hypertriglyceridemia, uh, and then your fats, uh, skeletal muscles will also eventually lead to decreased triglyceride clearance, and then your uh, it happens in fats and skeletal muscles. You'll have decreased triglyceride clearance, which will eventually lead to hyper, uh, basically high blood fat levels in the high levels of fat in the blood. Sorry, it's getting late and I'm like really sleepy right now. Okay, so I think the last few things are about the gut microbiome. Okay, the gut microbiome. It's colonized with bacteria on skin and the gut. We used bacteria products, and then we also support their growth. Gut microbiome help modulate and train the immune system. Uh, we also trade different products with them. They alter energy metabolism, and they can actually protect us against pathogens. They can be difficult to study because they're very hard to culture. Okay. The colonization of the GI tract occurs at birth, actually. And then vertical transmission from mother. Whoops, what did I do? There we go. Vertical transmission from of mother is dependent on the delivery. So whatever microbiome the mother had, it will be very similar to the child. And then this actually, whenever you have a C-section children, they actually have different gut composition than their mothers because it's not the same as the vertical transmission from normal birth. So it's another factor that helps us determine that because the C-sections have children have different gut mic composition than vertical transmission. Uh, it allows us to conclude that when given birth, a lot of the gut microbiome comes from the mother. Okay. The gut composition, so it actually varies between individuals. It depends a lot on the genetics and the environment. It remains stable over time, but actually, if you change your diet, you can completely change your entire gut microbiome. So diet affects it greatly. There's a lot of interactions between our intestines and the gut microbiome. I talked briefly about bile acids in the cholesterol. So we send bile acids, they get deconjugated by gut microbiomes, and then they send them back to us. We talked about short fatty acids. They help in the signaling, so those ones are involved. We talked about them briefly in, can't remember if it was fatty acid synthesis or fatty acid breakdown. And then urea cycle NH3 that allows interactions between the gut microbiome and the intestine. Carnitine being converted TMA that can also be exchanged between the gut microbiome and the intestines. Okay, production of short fatty acids by the gut microbiome. So the ones you make are the ones we'll talk about are acetate, propionate, and butyrate. So those are the short fatty acids, and they bind to G protein coupled receptors. You can make them differently. I don't think you guys are expected to know that. I'm pretty sure you're not expected to know that, but I'll just briefly talk about it. So you can convert pyruvate to lactate. Lactate can be converted to, I think this says acrylate, and then acrylate can be converted to propionate. Acetyl-CoA can be converted to acetate, and then you can run pyruvate acetyl-CoA, run a bunch of reactions, get butyryl-CoA, and then eventually convert butyryl-CoA to butyrate, and then you couple it with acetate going acetyl-CoA. Or you can also run two extra reactions down here, from butyryl-CoA to butyryl phosphate, and then from butyryl phosphate to butyrate. So you can get to butyryl-CoA to butyrate different ways. But yeah, you can make short fatty chain acids using the butyrate pathway, or you can use the propionate or acetate pathway. So you can make them differently. Okay. Signaling of short fatty acid synthesis. So remember, their blood concentrations is between 0.1 and 10 micromolars. Is that micro? No, that's milli. That's milli. That's not micro. So these are the short chain fatty acids. They bind through GPCR. So this is important because short chain fatty acids are made by the gut microbiome, and they have a lot of mechanisms inside our body. One thing is signaling. They're actually able to bind through G protein coupled receptors and mediate a lot of things. So they mediate blood pressure, immunity, the cell cycle, oncogenesis. So a lot of important stuff. And then they can also bind through non-GPCR signaling pathways. This will induce apoptosis, can inhibit HDAC, increase cell proliferation. So short chain fatty acids are very important that come from the microbiome because they have a lot of effects on our body. One of them is being signaling. Okay, so this is the uptake of them. They can use a transporter called SLC5A8. It couples it with sodium. It's a sodium coupled monocarboxylate transporter. There's also MCT1. You have uncharged bicarbonate exchanger, and you can also go through passive diffusion. So let's briefly talk about these. So this is your sodium potassium pump. This is calcium channels. This is chloride channels. So short chain fatty acids are here, and to get in, so this one in the picture, so this would be your uncharged HCO3 coupler, and then this would be MCT1. It doesn't show SLC in the picture, the one with the sodium coupled monocarboxylate transporter. But yeah, that's how you would exchange short fatty acids between our lumen and the bacterial cells. Not bacterial cells, the microbiome. Okay. Short chain fatty acids, so they're involved in signaling, they're involved a lot in energy homeostasis. So they have receptors on adipose, spleen, pancreas, intestines, and immune cells. They modulate glucose and lipid metabolism, and they eventually help normalize blood glucose levels. So this is your gut microbiome. You make short chain fatty acids. They travel to the intestines and then eventually go to the adipose tissue. They bind through GPCR. In adipose tissue, they decrease insulin sensitivity. So this is actually good when you decrease insulin sensitivity in adipose tissue, because now adipose tissue is less sensitive to insulin, so it won't take up as much glucose. So it won't make fats out of that, or it won't. So your fat accumulation eventually decreases. They can also act on muscle. They increase insulin sensitivity here. So insulin sensitivity in muscles is good. So that means the muscle is taking up the glucose from insulin, and then the muscle is able to contract and use that. And then they do the same thing with the liver, and then insulin sensitivity in the liver is always really good because your liver is the regulatory organ which helps regulate everything from blood glucose levels to glucose entering the blood or glucose leaving the blood. So you want to increase insulin sensitivity in the liver. So gut microbiomes and producing short chain fatty acids is very important in our body and our metabolism. Okay, so in summary, we talked about hormonal control, remember, of energy balance, remember insulin, glucagon, talked about briefly about epinephrine and how they bind. Receptor tyrosine kinase and signaling cascade of the end. We talked about GPCR, so know the difference between these two. AMPK, so that's different from receptor tyrosine kinase and GPCR, so it's activated by AMP. And then we briefly talked about the gut microbiome, so make sure you know all that stuff for this lecture. This was a long one, but I kind of did it pretty fast. So that's it for this video. I think I have one more to do. So thank you so much for watching and have a great day.